So hi, my name is Natália. Dr. Arias is my advisor. We are from the University of São Paulo, and I would like to talk to you today about part of the results of my PhD studies regarding the bivoltine behavior in the solitary Tetrapedia diversipes. So, bivoltinism is defined as the number of generation that is or broods that a specific species produces during 1 year period. So if there is one generation in 1 year, it's a univoltine species. If you have 2 generations, bivoltine, and so on. And we know that this kind of behavior is mainly influenced by many different environmental factors, but this behavior is also very important for the population structure and the population's social behavior. So one of the classical examples regarding how the bivoltine behavior may influence the social behavior in bees is in the bees Alictus rubicundus. So in these bees, these bees may be solitary or social, and when the females are solitary, they are univoltine, and when they are social, they are bivoltine. Because when they are bivoltine, the brood, you may find it's possible to find the generation overlap of the broods from the same female. So, just in more detail, in the generation 1 of this species, the founder will construct the nest, she will look for all the provision for that nest, and then the first brood will be biased for females, so you have 75% of these broods being females. And part of these females will follow the normal and regular process. They will leave the nest, they will mate, and then enter diapause to wait into the next generation to become founders as well. But part of the females will stay inside of the colony and will help the founders to take care of the brood in the next generation. So in the generation 2, all the brood raised will be raised with the help of the workers. And these broods will be male biased, and all the females produced will be founders as well. So they will leave the nest, mate, and wait until the next season to become founders. The bee I have used to study the bivoltine behavior is not social, so it's exclusively solitary bee. But this bee do have the bivoltine behavior and do have diapause in one of the generations as well. This tribe is distributed in the tropical region, but this specific species, Tetrapedia diversipes, we may found in some regions in São Paulo. So what happens in this species is that in generation 1, the female will construct the colony, find all provision for the brood, and then the brood will develop from egg to adult in only 1 month. But in the second generation, the female will do the same process to provide food for the brood, but the brood will develop much slower. So from egg to adult, they will take about 5 to 12 months to develop. And that's because in the last instar of larva development, in the 5th instar, the larva will enter diapause and will stop the development for a while. So my question for this work was, what is the difference between the founders from the first generation and the second generation regarding their transcriptome profile? So what genes are being expressed so these females produce different kind of broods? And for that, we have used the RNA-seq analysis. So all the RNA have been sequenced in HiSeq 2000 in the Illumina platform. And this species constructed the nests in holes in the wood. So it's nice because we can construct the strapped nests, and we will use that. So we have a spot in the University of São Paulo that we can just observe the bees there. And every time I realized that a female were constructing a nest, I just collect them while they are flying. All the collections have been made between 10 a.m. and 12 p.m., so could avoid a little bit of noise because of the period of the day. And also, I've pulled 3 females in each sample, and I have 3 samples from each generation, so for generation 1 and 2. So this is the base pipeline analysis. This is the pre-analysis, so after all the sequencing had been done, I just take the reads, make the quality check, quality trimming, and then digital normalization. This is the base pipeline for the assembly of the transcriptome itself, so everything that would be used to realign all the reads and make the counts. We have to do a de novo transcriptome assembly for these species, so we use the Trina to make this assembly. Then we have used the CTHIT program just to concatenate reads that were very similar. Then realign all the reads again with using bowtie 2, and then Chlorset to improve the transcripts assembly and the different isoforms assembly. And after that, we have made the counts, the differential expression analysis, and all the inferences regarding the transcripts. So, about the transcriptome itself, we have found for the females that were found in the colonies, concatenating everything from males from generation 1 into about 29,000 transcripts. These are the statistics for the quality of the transcriptome, so it was very good assembly. And for the gene expression itself, so we have found 36 transcripts that were differentially expressed in founders from generation 1 and founders from generation 2. Most of these transcripts, 34 of them, were up-expressed in founders of the generation 1, and only 2 of the transcripts have been up-expressed in founders of the generation 2. From all the 34 transcripts that we have found up-expressed in generation 1, only 4 of them have been annotated, and all these 4 transcripts, or these 4 genes, were related with mitochondrial and the respiratory chain. So this indicates that the females from generation 1 have higher energy demand than the founders in generation 2. And from the 2 transcripts expressed in generation 2, we could annotate one of them, and it's a SDR. Sorry. It's a SDR family member. This is a oxoreductase gene, and it's involved in many processes, so it's still not clear what is the influence in this process. But this gene is also differentially expressed in case differentiation in Apis mellifera, for example. So these are just to summarize the results, but what I would like to highlight at last is how important it is to study behavior with species that are not so modeled. So I know there's a number of problems that we can find in making transcriptome assemblies, but we also know that the database are very biased for model species. So many of the transcripts and many of the things we find we cannot annotate because there are no representing for that. And as in this case, for example, most of the transcripts I could not find in the gene ontology or any other database, so they may be important for this important behavior in this piece. And just would like to thank you very much for everybody for being here, for the people in my lab, and for the organization for giving the opportunity to be here.
We have plenty of time for questions. Questions, anyone?